This presentation will be over differential expression of genes between cystic fibrosis and healthy individuals. It was made and presented by Mariah, Ainsley, Jianing, and myself. Our problem was over cystic fibrosis is a debilitating and thus far an incurable disease. Our question is, which genes are differentially expressed in individuals with cystic fibrosis? For our hypothesis, the gene is differentially expressed between healthy individuals and individuals with CF. Our objective, to determine the most differentially expressed genes in individuals with CF and investigate current therapy treatments. Cystic fibrosis manifests as a lung ailment characterized by mucus obstruction and chronic airway inflammation. It stems from the mutations in the CFTR gene. Despite advancements in therapeutic approaches, treatment efficiency varies due to the diversity of CFTR gene mutations among individuals. This project aims to inspect the genetic differences between CF patients and healthy individuals, leading to further understanding of treatment strategies. To help investigate different gene expression associated with CF, resources such as NCBI Gene Search, GEOTR, and the ASU library will all be utilized. Insights on cystic fibrosis and gene expression will support the therapeutic interventions not only now, but for the future as well.
Some background information about cystic fibrosis is that it is an inherited disease caused by a mutation in a gene called CFTR, and this gene provides instruction for the CFTR proteins. The CF patients with defective proteins will affect their body cells, tissues, and glands that make mucus and sweat, and the CF causes the mucus to be more sticky and block the pathway, especially in the lung and pancreas, leading to severe damage or infection in the lungs and digestive system and other organs in the body. The symptoms and severity can vary widely among individuals with the CF, and some people can have few or no symptoms, while others experience severe symptoms or life-threatening complications. To test our hypothesis and find genes that may contribute to CF, we utilize several databases.
We used the GSE 38-67 dataset from a research paper by Babu and Nobel for investigating the intersection of expressed genes associated with COVID-19, cystic fibrosis, and kidney disease. This dataset was put into GeoToR, and we created two groups consisting of healthy individuals and individuals with CF. You can see from the scatter plots here that there is a significant difference in the expression of different genes between healthy patients and those with cystic fibrosis. For this project, we selected the top 5 genes more highly expressed in cystic fibrosis individuals, which were the genes found by GeoToR that had the smallest p-values, highest high t-test score, and highest positive log-FC score, as a negative log-FC score would have meant the gene was more expressed in healthy individuals. So all of our genes here have a p-value of less than 0.05. In the center here are the 5 most differentially expressed genes from the data set. We took each and searched each in the NCBI gene data bank by their GenBank code given by the GeoToR to get the gene name and any additional information. So the first gene interacts with the CFTR gene, and when we looked at tissue expression, we found that it's primarily expressed in the testes and is linked to male infertility in cystic fibrosis patients. The second gene, CLEC4D, acts as a pattern recognition receptor for the innate immune system, and some research suggests that it may contribute to increased lung infections and inflammation in cystic fibrosis patients. The third gene displays activity toward mucin-derived peptide substrates and is expressed primarily in the kidney and whole blood. The fourth gene codes for voltage-dependent calcium channels and is expressed primarily in the brain, but there are not very many research articles linking it to CF. The fifth gene is expressed in the colon, liver, and whole blood, and some research suggests that it can affect liver biochemistry and may contribute to liver disease in cystic fibrosis patients.
Through our analysis of gene expression patterns, we identified the top 5 DEGs associated with the disease. Our study revealed significant transcript levels in the colon, small intestine, stomach, liver, and the lungs. When comparing gene expression profiles between CF patients and healthy individuals, we identified DEGs with log-fold changes, and those with p-values less than 0.05 were considered significant. Our findings supported our initial hypothesis and align with the lung and digestive symptoms commonly seen with the disease. Our research also opens up possibilities for potential gene therapies targeting DEGs and CFTR genes. This could lead to improved management of CF and potentially other hereditary diseases. Our research is particularly significant given the few therapeutic studies available for CF. However, our research does not account for other factors influencing gene expression, such as environmental or epigenetic factors.